Okay, so first of all, I want to thank the organizers for giving me this opportunity. I'm a collaborative postdoc fellow between the Dan Fletcher's lab at UC Berkeley and the BioHub. I'm going to tell you a story about how influenza A viruses achieve directed motion without having any internal motors. So respiratory viruses like influenza must cross extracellular barriers such as the mucus layer. So here I'm showing you a cross-section of tracheobronchial epithelial cells, which have been stained for glycoproteins. As you can appreciate from this image, this mucus barrier is quite thick, around 50 microns, but it also has this increasing density of glycoproteins as you get closer to the cell surface. Adding another layer of complexity is ciliary flows generated by the beating of cilia. And that causes this layer to be continuously cleared. And in humans, that time scale is around 20 minutes. So an influenza virion, and they have quite a wide size distribution. They can range from 300 nanometers all the way up to a few microns. So they must cross this barrier within a time scale of 20 minutes on average. If you assume simple diffusion, you'd get a probability of infection that's very small, and we would never get the flu. So what strategies do influenza viruses employ to transit host barriers? So one strategy was maybe discovered in Dan Fletcher's lab a few years ago. So clinical IAV isolates are filamentous. And what they found is that there's this spatial segregation of proteins on their surface. So shown here in blue is hemagglutinin, HA, that binds to sialic acid receptors abundant in mucus. NA, or neuraminidase, is an enzyme shown in red that's spatially confined to the pole, and it enzymatically cleaves these sialic acid receptors. So when they place these virions on a cover slip functionalized with sialic acid, what they found is that these virions persistently stepped away from their neuraminidase-rich pole. So I took the data from these trajectories, and I did a mean square displacement analysis. So for the polarized virions, that revealed a super diffusion with an exponent of 1.5, indicating a self-avoiding walk. But interestingly, in the same paper, they also did a perturbation where the neuraminidase is no longer tethered to the pole, so it's more uniformly mixed. Even in that case, the slope is around 1.5. However, the superdiffusion persists for much longer in this wild type versus the mutant. And that's you can see that in this visualization of the experimental trajectories, where the polarized trajectories travel further and can explore more space. So how do we explain this? So late last year, we published a model, a 1D model that explains how they achieve this directed motion. So we modeled the virions as a rigid elongated shell placed on top of a 1D substrate of sialic acid receptors. The sialic acid can bind reversibly to hemagglutinin. And we modeled this, so these are connected to glycoproteins, and so which have a finite extensibility. And so once bound, they can exert a force and cause the virion to move either to the left or to the right. Neuraminidase can irreversibly cleave or hydrolyze these receptors, generating energy for the locomotion. Without going into too many details, we did stochastic simulations using an overdamped equation of motion that couples chemical kinetics with motion. But we also had a mean field description that balances the free energy production due to neuraminidase with dissipation that includes bond friction and viscous damping. So the output of these models is a characteristic speed of migration. And so the main takeaway from this work was if we now plot the mean speed as a function of the hemagglutinin binding affinity on the x-axis and the enzymatic activity on the y-axis, compared to the mixed distribution of HANA, which is confined to this sweet spot, you get a much larger expanded Goldilocks zone in binding affinity for these polarized virions. which is largely insensitive to neuraminidase activity. And this might explain why neuraminidase inhibitors like Tamiflu may not be good at blocking initial infection. So then I took data from human-type influenza strains binding to human-type receptors from independent experiments, and I plotted where they cluster, and they cluster very nicely on this predicted zone for locomotion. Now, if you take avian strains binding to human-type receptors, they cluster outside it. So this 1D model explains how influenza viruses harness their environment as an energy source to move, to get to the cell surface. But what about in 2D, where you can have now rotational degree of freedom and trajectories can self-intersect, for example? So, and we can ask questions such as, what is the role of HA to NA stoichiometry? Like, what happens if you have more NA versus more HA, for example? So these are, so we extended the 1D model to 2D now. So these are example trajectories for polarized virions versus mixed. And these are multiple independent runs on a 2D uniform substrate. And what you see is that the blue, which is the polarized virions, they cover a much larger distance. And if we zoom into one trajectory, you see that the virion moves persistently away from the neuraminidase-rich pole, cleaving substrate sites, and the track is quite persistent. Compare that to the mixed case, where you have quite random motion, and the virion can self-trap or get trapped within its depleted sialic acid tracks. So, and these trajectories are pretty similar to what they saw in experiments. So then I took, so then I can do the same MSD analysis, and just like experiments, the simulations recapitulate the exponent, which is around 1.5, but they also tell us that the uniform case gets trapped, and the superdiffusion doesn't persist for as long as for the polarized case. So what else can the simulations tell us? So here, using infection rate as my metric, because that's ultimately what we care about, how fast do these virions cross a certain threshold? Using that as my metric, I plot on the x-axis the HA to NA stoichiometry. So on the left, you have more NA. On the right, you have more HA. And then the enzymatic activity on the y-axis. What we see first is with increasing enzymatic activity, you have more infection. But there's an optimum in like the molecular architecture that maximizes infection. So it seems that around 1-to-1 or 2-to-1 HA to NA maximizes space exploration. And that's because if you have too much HA, the trajectories have, the HA has more opportunities to bind and cause decorrelation of the track, so you get more wiggliness. If you don't have enough HA, you don't have enough sampling of the environment or the required force for propulsion. But this was assuming a uniform receptor density, so uniform sialic acid receptor density. But what happens if that's not true? What if there's a gradient now? So in this case, there's increasing sialic acid density as you go down. And what happens if we run the same kind of simulation? So you'll see that the trajectories are biased towards higher sialic acid density regions. And that you might think that maybe a mechanism by which they can detect where the cell surface is, and they know where to infect. So then if I do the same kind of plot with the infection rate as heat maps, we see that there's like these, the infection rate, first of all, without gradient compared to with gradient increases by a lot. But there's like this population here that's around 1-to-1 or 2-to-1 HA to NA. That's good at exploration, but also at reading the environment. And that it does that by segregating this energy generation with the reversible sampling of the environment. So the idea is by this segregation, you can both read the environment, but also move. But curiously, there's this other population that emerges here. So that means like even without any cleaving activity, without neuraminidase, you can sort of detect where there's more stuff. So you do just by reversible binding, unbinding with receptors. So, and this actually mirrors the bimodal distribution seen in natural IAV populations in experiments. So you have this population here, some fraction, which are termed explorers, that have like a 1-to-1, 2-to-1 distribution of HA and A. And you have this population that's like 10-to-1 HA2NA that are termed sensors. So could this be a barrier crossing strategy that IAV exploits as a population rather than just random noise? And that's something I'm currently working on. OK, so how do we test all of this? So I've been working on this experimental setup where I can test some of these hypotheses using supported lipid bilayers that have been functionalized with sialylated proteins. And I can force them to cluster, and then I can see how influenza virions respond to those heterogeneities. So I'm tracking the virion motion in 2D using TURF, and then also simultaneously tracking the sialic acid receptor distribution. So here are a couple of movies where the sialic acid intensity is in orange, and these are lab strain influenza virions, which are spherical. And even these spherical strains sort of like go towards these clusters of sialic acid density, intensity, higher intensities. So if I take data from from these experiments and correlate motion with the intensity of sialic acid, I see that there's a systematic bias, even for these spherical virions, towards higher sialic acid intensity. So to summarize, the 1D model explains how these virions hijack the environment, the extracellular environment. And the 2D model explains how the trajectories are persistence for a given molecular architecture, and this idea of distributed transport strategy. I've also developed an in vitro assay that can help test some of these hypotheses. So this opens up a few exciting questions that we're interested in. So for example, can we modulate the hemagglutinin binding affinity to block infection? Or can we manipulate sialic acid gradients in mucus to block infection? It also suggests that maybe we can use motion on, let's say, cover slips as a diagnostic of the type of IAV strain. And then sort of the overarching question that I'm interested in is, like, these were single virions. What happens at the population level? How do they exploit the environment? So I want to thank Dan Fletcher, Greg, Boris, and then Sandy, Amy, and Carolina for all the funding, support, and constant feedback, and everyone in the Fletcher lab. Thank you so much.
Great talk. Thank you. All right, we have time for maybe one question. Let's see right here.
Hey, really cool talk. I was curious if there is an asymmetry that is orthogonal to the main axis of asymmetry. I guess theoretically, I would expect them to start going in circles. Do you ever see that in your data?
Yeah, so I think in the experimental data, we see that these virions, they don't have like the mixed case. It's not uniformly mixed. There's some asymmetry in that direction. So then they start going like, they get trapped basically.
So, you know, I noticed in your plots you had log axis, but then the infection rate is linear.
Yeah.
And how do I think about like a factor of 2 change in the infection rate? Maybe I don't understand quite how the infection rate is.
So I think you have to convert that into probabilities. So that's like 1 minus exponential times the rate times the time. So then, yeah.
Okay. Let's thank our speakers for this session. Those were all awesome talks. Thank you so much.